Hi, in today's session we're going to be answering an exam style question on protein synthesis for the WJEC A Level Biology. So this is question 1, which I'm doing for protein synthesis. So without further ado, let's get right into it. So this is the question that we have. Describe how a nucleotide sequence on a DNA molecule results in the production of a polypeptide. So as you can see, I've highlighted polypeptide here because we're going to be talking about protein synthesis. And when this question came up, this was worth 10 marks. So it's an essay style question, so we need to have enough points in order to get full marks for this question. And if you are wondering, this is taken from the WJEC Biology June 2013 paper. So let's see how we're going to approach to answer this question. Now, before I even jump straight into the mechanism for polypeptide chain production, I'm just going to give a brief overview about what DNA actually is. So I'm just going to switch to blue, and what I'm going to first say is that we know that the nucleotide base sequence of DNA we know that codes for the amino acid sequence. So that fundamentally summarizes what DNA actually is before we actually jump into how it actually works. So the nucleotide base sequence of DNA codes for the amino acid sequence. Okay, so let's just give some more detail about DNA. So obviously we know DNA is got DNA. We know it's got 4 types of bases, and just to say what that is, so that would be adenine, thymine, cytosine, and guanine. And from the properties of DNA, we know that 3 bases code for 1 amino acid. So I'm just going to write that down here. 3 bases equals 1 amino acid. That's going to be quite important when we talk about codons in translation. So that's quite important property of DNA. So I'm just going to make sure we know that's a DNA on the DNA properties. Okay, so we're first going to talk about transcription. So I'm just going to put transcription down. So let's give a brief overview about what transcription is now. So transcription. So this is stage 1 of protein synthesis. So this is where we basically produce a mRNA molecule that carries the genetic code, or you could just say info for making proteins. It's obviously very important for DNA to make mRNA, because DNA, we know that's in the nucleus. DNA is so large that it can't actually leave the nuclear pore itself, so it actually needs to make an mRNA molecule. Basically, you could think of it as a shorter version of itself, which can easily be passed through the nuclear pore and first result in the production of protein on the ribosomes, which we'll come on to later. Alright, so that's an overview of transcription. So let's start. Now let's just explain what transcription is for this first stage of protein synthesis. So we've got DNA basically in its 2 polynucleotide strands. So we're going to use an enzyme, DNA Helicase, to essentially unwind the DNA. commas, apostrophe, sorry, to just to highlight that word there. Unwind DNA to expose its bases. And the way this works is that DNA helicase breaks the hydrogen bonds between the complementary bases. So that's very important. That's the first stage in transcription, is to actually break the DNA. So, alright, so now once we've broken the DNA, so we've got the strands separated now, the important thing to understand with transcription is, now one of the strands will be used as the parental strand, or coding strand. I should say as the coding strand. So the key part to understand that it's one, it's not both of the strands that we separated, it's just one, which is the opposite in when you have DNA replication where you use both strands. This is in protein synthesis, you're just using one of the strands, and that's very important to understand and to actually make sure the examiner knows that too. So one of the strands will be used as a coding strand. Right. So once we've got our strand now, we're going to introduce another enzyme called Rna Polymerase. So RNA polymerase, this is basically going to link to the DNA. Or to be more specific, to the start of the coding strand. So we've got this enzyme called RNA polymerase attached to the start of this coding strand. So what's going to happen now is that there's going to be in this region in the nucleus, there's going to be 3 RNA nucleotides. Three RNA nucleotides basically align opposite to the complementary base. So for example, so if you've got on the DNA molecule, let's say you've got on the DNA, you've got let's say for argument's sake CCG, then the RNA nucleotide in this case, because it's complementary base pairing, that's going to be, I'll do some different colour just to make this really important. This would be GGC. So you can see the complementary nature here. So this is going to basically keep repeating. So the RNA nucleotide, so that enzyme, which is basically allowing all this linking to happen, is going to keep moving along the coding strand. So that keeps moving along the coding strand. Until the mRNA molecule is synthesized. So once you have the mRNA ready, so these basically all these nucleotides joined together because of the actions of RNA nucleotide, then the RNA nucleotide is gonna detach off the coding strand. So that's gonna basically release itself now. So that's done. We've got the mRNA. So the next step between translation and transcription and translation is splicing. So I'm now going to explain how we're going to approach this step. So imagine we've got the mRNA here, so that's actually pre-mRNA. So you've got these sections here. I'm going to just highlight one of this like this. So these are the ones which aren't coloured in. These are exons, and this is introns. So these are coding. So this is going to actually code for the protein. This is non-coding for protein. So the next step actually is to remove this by a process called splicing, so that you remove the introns and you essentially just have the exons left now. So this is just exons. So this is the so this is what we're going to explain next. So to put it in words in the exam, what you would say is that pre-mRNA contains introns and exons. Yeah. Okay, let's do a bit better the S there. Exons. So therefore, by process of splicing, exons, or I should say introns, are removed. So that produces your functional mRNA now, which is actually going to go to the ribosomes. So this is all taking place in the nucleus. So this mRNA is going to leave for the nuclear pore. So leaves nucleus through nuclear pore. So that's how it actually leaves the nucleus. So that's it. So we've got the mRNA, we've removed the introns, and now it's ready to provide the genetic information and code to make the protein. So that's what we're going to explain now. So translation. So getting right into it now. So the mRNA, what it's going to do is going to attach to the ribosomes. Which are actually, as we know, they're on the rough endoplasmic reticulum. So I'm just going to write RER there for short. So just to explain a bit about the structure we're talking about now. So obviously ribosomes, we know that's got, in terms of like its actual structure, it's got a larger subunit and a smaller subunit. So it's the larger subunit, that's where you have the tRNA binding site. And the smaller subunit is where the mRNA attaches. Right. So now we mentioned the word tRNA, so we should explain what tRNA is now. So basically tRNA carries the anticodon for a specific amino acid. So now we've explained all the parts now in translation. So we've explained what ribosomes are, where they're located, its structure, and also tRNA. So now we'll also explain the mechanism of translation. So what we're going to do is we're going to say the mRNA attaches to start of ribosome. So there we are. We've got the mRNA on the ribosome, so we're ready for protein synthesis. So now the tRNA, so I'm going to put in bracket just to remind the examiner and ourselves in the exam, carrying the specific amino acid binds to the codon on mRNA. So to basically form a codon anticodon complex. So once that tRNA has binded to the mRNA, on the adjacent side you're going to have amino acids forming. So I'm just going to write down words now. So on the adjacent side of tRNA, amino acids bond together through peptide bonds. So once the amino acid has been attached to the growing polypeptide chain, so once amino acid attached, the tRNA is basically going to leave the complex and pick up another amino acid. So tRNA picks up another amino acid, which this process requires ATP, so we're just going to make that clear to the examiner. It requires ATP. And then this will basically bring that another copy of that amino acid back now. So therefore, this will bring back another copy of that specific amino acid. And so this process is continually taking place. So what we need to now say is that this process continues to take place until you basically reach the stop codon on the mRNA. So once you get those 3 bases on the mRNA, that's when protein synthesis will stop, and then that's your polypeptide chain complete. To take place until, so we're going to say here, until the, instead of end we'll say stop codon. Reached on mRNA. So once we've reached the stop codon, therefore polypeptide chain has been synthesized. And synthesized is basically mean it's been produced now. Okay, that's it. That's 10 marks. So you can see here we've got enough detail So if we just go from the start here. So we started by talking about an overview about what DNA actually is. Then we moved on to transcription, about producing that initial mRNA molecule that contains the genetic code for producing the protein, polypeptide chain. Then we've talked about splicing, introns and exons. And then we've talked about translation. So I hope that was helpful on this topic of protein synthesis. If you have any topics that you would like me to cover, then leave them in the comments below. Other than that, I hope you have a nice day.